Okay, what you're looking at is where I work, the Salk Institute, looking out over the Pacific Ocean. And I'm going to be telling you today about dynamical differential covariance, a new method for functional connectivity. And this is primarily the work of graduate student in my lab, UC Chen, who's, as you can see in the inset. So first of all, if you look at the brain, there's the spatial scales range over 10 orders of magnitude from the molecular layer at the bottom to looking at the global patterns of activity, which this group, I think, is particularly interested in. And, you know, there's dynamics occurring at each one of these levels, and it's really quite remarkable. And the real challenge is to try to bridge these levels. So the particular approach that I'm going to take is a way for analyzing data. The tremendous amount of deluge of data is now being poured out from labs, from recordings from not hundreds of thousands of neurons simultaneously, and also from large-scale recordings from the cortex and other parts of the brain. And the method I'm going to tell you about can be used for each one of these levels, basically. It's a very general approach. So some of those popular methods for analyzing functional connectivity are statistical, and perhaps the most popular is covariance, which tells you something about how two areas of the brain may covary in time. And we all know that if two areas get a common input, they can covary without having a direct connection, which is a confound. Now you can still learn something from it. Why? Well, if you look at the resting state, which has been analyzed with independent component analysis, you can learn something about the fact that two areas covary, even if there's no direct connections between them, because of the fact that they're being driven simultaneously. So, but if you are interested in the connectivity, then one way to reduce those confounds is to take the partial covariance, the inverse of the covariance, and that is effective because you're using conditional correlations. Now, a couple of years ago, we introduced yet another statistical measure, which we call differential covariance, where instead of correlating the signals at two areas, you correlate the derivative of the signal with the signal at two areas. And the intuition behind this is that if you have two neurons that are connected, one which is the source, and the axon goes and produces a synapse on another neuron in another area, the current going in is proportional to the derivative, the change in the voltage. And so the idea is that you might be able to get an idea between to get directionality. So none of these measures, the covariance or the partial covariance, give you directional information because they're symmetric. But this measure is not symmetric. So just to see if it works, we put together a simple network consisting of 50 model neurons. Details are not important, except to say that here's the connectivity diagram. And you can see that the neurons were connected with the spacing of 10 in a one-dimensional circle. And here's the blow up, so you can see what it looks like. Now we're going to compare the covariance. This is what the covariance looks like. You can see that it does not pick up the connectivity. And the precision matrix, which is another name for the partial covariance, is also doesn't have, it picks up something. Actually, you can see something here, but it's not the ground truth. But the part is this is now the dynamic the differential covariance, which I mentioned previously. It picks it up beautifully, and you can actually see that it picks up with different signs, both there's the sender and the receiver. So in principle, it might be able to be useful. But okay, very recently, those are all, by the way, ad hoc. I mean, they would just put together as statistical measures as that maybe they'll help. Okay, so, but really, I think the gold standard here should be to, this is something that Carl has pioneered, is to use a model. Start with a model and then have it model based. Now, the trouble there is finding the right model, and that can be very computationally complex problem. And we stumbled on a very simple model, which actually turned out to be very, very insightful for understanding some of the statistical measures. Okay, so here's the world's simplest neural model. Everybody, who starts out with a model in which you have the time derivative of the membrane potential equal to a connection matrix W times the activity, right? And so this is a linear model, but let's just do a little statistical analysis. Let's take the outer product with the activity and average over time. Well, here's what you get. Isn't it interesting? This is the differential covariance, and this is the covariance. And if you solve for W to get an estimate for the connectivity, you get two terms. You get the differential covariance and you get the partial covariance. In other words, you take these two measures that were developed, for other purposes, and multiply them, you actually might get a pretty good estimate. And by the way, and with roughly the same cost as the covariance. The covariance is very fast and cheap to compute. So this is basically two forms of covariance that are multiplied. And this is published, by the way, just last week in PNAS. So if anybody's interested, just go to PNAS and take a look at the details. Now, what we showed mathematically was that, first of all, it's unbiased. This approach is unbiased, and it also is, it works for non-stationary signals. You don't have to assume that it's stationary when you're averaging, which a lot of people do, of course, with the covariance method. Now, this is not one method. It's actually a class of models, because you can replace the linearity with a nonlinearity, right? So R can be a sigmoid, for example, or it could be a ReLU, or which is very popular now in deep learning, which is when you have a sharp threshold, and then if you put that into the equations, it just follows right through. And the only difference now is that for the partial covariance, you're using R, and that's very simple to compute. Okay, well, let's try it out on our confounding examples, and this is the one here in the lower left-hand corner. And what you can see is that when you apply it, if you put in a linear equation or a nonlinear equation, that the 2, 1 and the 3, 1, which are the true connections, come out, and you don't get 3, 2, right? 3, 2 does not is not it actually picks out the correct connectivity, and it does that for chains, and it also does that, interestingly, for chaotic systems. This is the classic Rossler system, and as you can see, x1 only depends on x2. And there you go, it picked it out. X2 only depends on X1, and that's also here. And then X3 only depends on X1. So again, we're picking out the correct connectivity in terms of the equations for a nonlinear chaotic system. Okay, we wanted to try it out on other ground truths, and we put together a very simple Leaky integrating fire. Now this is applying it to single neuron models, and you can see that you get nice spikes for a couple of neurons, and we applied a wide range of methods, covariance methods, various sorts, and then here's our new dynamic differential covariance methods. Although you can distinguish them by this delta. The delta is the differential dynamic models with either covariance or precision or the full-blown dynamic equations. And the Granger. Now here's what's interesting is that this is now computing the area under the curve. And here are the curves up here in the upper right. But you can see that these covariance methods really don't get off the ground. These are the controls. But the differential covariance methods do quite well. And in fact, the Granger method does extremely well. Very impressive. This is a traditional method which is used. It's a linear model and it's an autoregressive model. However, now let's look at the time it takes to compute the answer, and this is a log scale. So all these covariance methods are down here in the second range, right, below a second. But the Granger method is way up here, it's 60 hours. I mean, it's unbelievably time consuming. This is well known. Okay, well, that's good. It can be used for spiking models. What about these neural mass models? Well, here's one. This is macroscopic, the Wong-Wang model. And we put in the connectivity from the Human Connectome project. And here we go. We can show that, compared to the controls, in fact, a lot of these methods do pretty well. Precision matrix and Granger. But in fact, our simple dynamic model is actually doing the best. So we applied it to fMRI data, and this is from the Human Genome Project, and resting state data. And this is a wonderful data set. There's 1,000 individuals, so we basically analyze each one separately. And what we did was, first of all, we started with the independent components, the ICs, and looking at this, particularly the ones that are the most prominent, and then we compared across individuals and looked for, and this is a histogram of all of the components that were the ones that were the most significant, p less than 0.01. And here's the threshold. We can vary the threshold, but this threshold is set so that 90% of all the individuals had this component from the resting state. And so here are just a handful of them, and you can see that there are some that are in the occipital region. There are some that are at the parietal region and the prefrontal region. So we can pick out some of the strongest connections, the backbone connections, we call them. And we can compare them actually with diffusion MRI. This is the track tracing. This is the Human Connectome project. And this shows you the comparison between the connectome, presumably, I always thought of them as ground truth, but from what I've heard, it's maybe not quite as ground as we had expected or hoped. But in any case, it's probably the strongest ones are probably accurate, and those are the ones that we're going for. And we're going to change the binarization p-value, right? We changed, we picked out p less than 0.01, but we can look at more and more if we reduce the p. And you can see here that just the covariance and the partial covariance, and they all agree better, of course, as you make it more stringent, you have fewer components that meet the criteria. But all of the differential methods up here, dynamic differential methods, are actually superior, and you can see that they agree better as you get the binarization threshold goes up, so by a factor of 2 or more. So the same thing, so this is the actual proportion of strong diffusion connections. Okay, well, that's my 10 minutes, but let me just summarize. So first of all, this is a method that gives unbiased directional estimates of W, which can be very useful if you're interested in causality. DDC can decrease the systematic inference error due to model mismatch. It's very forgiving. Our model, even a simple linear model, actually works extremely well with the fMRI data. DDC is robust to added Brownian motion noise. I didn't show you this, but you can add a lot of noise and it still is able to give you good results. And that you can use it for analyzing non-stationary data whose higher order statistics vary with time. That is to say, a little window that you move along, and you can see as it changes, you'll be able to pick out the connectivity, despite the fact that the covariance structure is changing. Now, finally, and this is for video people, I'm going to give you a tachistoscopic quick run through for recent papers for those who are interested. This one is accepted but not yet published. It's in Network Neuroscience. And this is where we've applied these differential methods to fMRI data and compared it with behavioral reaction times. We can predict reaction times. Reaction times are really interesting because it's been shown that they're actually correlated with this intelligence value, that this big R, which is supposed to be a global indicator of intelligence and with intelligence tests. And so if this is true, then this may be a way of tapping into that, this one global factor. And here's a paper from Chaos. This is the delayed differential analysis I was telling you about in the question period in the last talk, which is it's an interesting way of getting causal structure. And what it does is we call it dynamical ergodicity in Y. And the reason is that we have two terms. The real problem with causality measures is that if you have two signals that are synchronized, they may be synchronized for no causal reasons. That is to say, they're getting a common input or some other reason. And so this is a way of eliminating those situations. And finally, I mentioned this as well, that you can use these DDA methods to pick out chimeras and other nonlinear oscillator phenomenon in a very nice way. So thank you. All the software, DDC software that I presented is going to be available on GitHub. It's very easy to use. I mean, these are very, like, if you've been using Covariance, this is basically plug and play. It's not like it requires a tremendous amount of complexity. DDA does require, there's like 4 or 5 flavors, and it does require that you know something about dynamical systems theory. So that's the end of my talk. And I will stop sharing.
Thank you. Again, brilliant. Thanks so much. Just a little comment before I invite Pedro to ask his question.